Human Genome Project. In the preceding sections, you have learned that it is the sequence of bases in DNA that determines the genetic information of a given organism. In other words, genetic makeup of an organism or an individual lies in the DNA sequences. If two individuals differ, then their DNA sequences should also be different, at least at some places. These assumptions led to the quest of finding out the complete DNA sequence of human genome. With the establishment of genetic engineering techniques where it was possible to isolate and clone any piece of DNA, and availability of simple and fast techniques for determining DNA sequences, a very ambitious project of sequencing human genome was launched in the year 1990. Human Genome Project, HGP, was called a mega project. You can imagine the magnitude and the requirements for the project if we simply define the aims of the project as follows. Human genome is said to have approximately 3 × 10⁻⁹ base pairs, and if the cost of sequencing required is $3 per base pair, the estimated cost in the beginning, the total estimated cost of the project would be approximately $9 billion. Further, if the obtained sequences were to be stored in typed form in books, and if each page of the book contained 1,000 letters and each book contained 1,000 pages, then 30,300 such books would be required to store the information of DNA sequence from a single human cell. The enormous amount of data expected to be generated also necessitated the use of high-speed computational devices for data storage and retrieval and analysis. HGP was closely associated with the rapid development of a new area in biology called bioinformatics. Goals of HGP. Some of the important goals of HGP were as follows. 1. Identify all the approximately 20,000 to 25,000 genes in human DNA. 2. Determine the sequences of the 3 billion chemical base pairs that make up human DNA. Store this information in databases. 4. Improve tools for data analysis. 5. Transfer related technologies to other sectors such as industries. Address the ethical, legal, and social issues, ELSI, that may arise from the project. The Human Genome Project was a 13-year project coordinated by the U.S. Department of Energy and the National Institute of Health. During the early years of the HGP, the Wellcome Trust UK became a major partner. Additional contributions came from Japan, France, Germany, China, and others. The project was completed in 2003. Knowledge about the effects of DNA. Variations among individuals can lead to revolutionary new ways to diagnose, treat, and someday prevent the thousands of disorders that affect human beings. Besides providing clues to understanding human biology, learning about non-human organisms, DNA sequences can lead to an understanding of their natural capabilities that can be applied towards solving challenges in healthcare, agriculture, energy production, environmental remediation. Many non-human model organisms such as bacteria, yeast, Caenorhabditis elegans, a free-living non-pathogenic nematode, Drosophila the fruit fly, plants, rice, and Arabidopsis, etc., have also been sequenced. Methodologies. The methods involve two major approaches. One approach focused on identifying all the genes that are expressed as RNA, referred to as Express Sequence Tags, ESTS. The other took the blind approach of simply sequencing the whole set of genome that contained all the coding and non-coding sequence, and later assigning different regions in the sequence with functions, a term referred to as sequence annotation. For sequencing, the total DNA from a cell is isolated and converted into random fragments of relatively smaller sizes. Recall DNA is a very long polymer, and there are technical limitations in sequencing very long pieces of DNA and cloned in suitable hosts using specialized vectors. The cloning resulted into amplification of each piece of DNA fragment so that it subsequently could be sequenced with ease. The commonly used hosts were bacteria and yeast. and the vectors were called as bacterial artificial chromosomes and yeast artificial chromosomes. The fragments were sequenced using automated DNA sequencers that worked on the principle of a method developed by Frederick Sanger. Remember, Sanger is also credited for developing method for determination of amino acid sequences in proteins. These sequences were then arranged based on some overlapping regions present in them. This required generation of overlapping fragments for sequencing. Alignment of these sequences was humanly not possible. Therefore, specialized computer-based programs were developed. These sequences were subsequently annotated and were assigned to each chromosome. The sequence of chromosome was completed only in May 2006. This was the last of 24 human chromosomes, 22 autosomes and X and Y to be sequenced. Another challenging task was assigning the genetic and physical maps on the genome. This was generated using information on polymorphism of restriction endonuclease recognition sites and some repetitive DNA sequences known as microsatellites. One of the applications of polymorphism and repetitive DNA sequences shall be explained in next section of DNA. Fingerprinting. Salient features of human genome. Some of the salient observations drawn from Human Genome Project are as follows. 1. The human genome contains 3,164.7 million base pair. 2. The average gene consists of 3,000 bases, but sizes vary greatly, with the largest known human gene being dystrophin at 2.4 million bases. 3. The total number of genes is estimated at 30,000, much lower than previous estimates of 80,000 to 140,000 genes. Almost all 99.9% nucleotide bases are exactly the same in all people. 4. The functions are unknown for over 50% of the discovered genes. 5. Less than 2% of the genome codes for proteins. 6. Repeated sequences make up very large portion of the human genome. 7. Repetitive sequences are stretches of DNA. Sequences that are repeated many times, sometimes 100 to 1,000 times. They are thought to have no direct coding functions, but they shed light on chromosome structure, dynamics, and evolution. 8. Chromosome 1 has most genes 2,968, and the Y has the fewest 231. 9. Scientists have identified about 1.4 million locations where single-base DNA differences SNPs, single nucleotide polymorphism, pronounced as SNPs, occur in humans. This information promises to revolutionize the processes of finding chromosomal locations for disease-associated sequences and tracing human history. Applications and future challenges. Deriving meaningful knowledge from the DNA sequences will define research through the coming decades, leading to our understanding of biological systems. This enormous task will require the expertise and creativity of tens of thousands of scientists from varied disciplines in both the public and private sectors worldwide. One of the greatest impacts of having the HG sequence may well be enabling a radically new approach to biological research. In the past, researchers studied one or a few genes at a time. With whole genome sequences and new high-throughput technologies, we can approach questions systematically and on a much broader scale. They can study all the genes in a genome, for example, all the transcripts in a particular tissue or organ or tumor, or how tens of thousands of genes and proteins work together in interconnected networks to orchestrate the chemistry of life. DNA fingerprinting. As stated in the preceding section, 99.9% of base sequence among humans is the same. Assuming human genome as 3 into 10 to the power 9 base pair, in how many base sequences would there be differences? It is these differences in sequence of DNA which make every individual unique in their phenotypic appearance. If one aims to find out genetic differences between two individuals or among individuals of a population, sequencing the DNA every time would be a daunting and expensive task, Imagine trying to compare two sets of 3 into 10 to the power 6 base pairs. DNA. Fingerprinting is a very quick way to compare the DNA sequences of any two individuals. DNA. Fingerprinting involves identifying differences in some specific regions in DNA, sequence called as repetitive DNA. Because in these sequences, a small stretch of DNA is repeated many times. These repetitive DNA are separated from bulk genomic DNA as different peaks during density gradient centrifugation. The bulk DNA forms a major peak, and the other small peaks are referred to as satellite DNA. Depending on base composition, A is to T-rich or G is to C-rich, length of segment, and number of repetitive units. The satellite DNA is classified into many categories such as microsatellites, mini-satellites, etc. These sequences normally do not code for any proteins, but they form a large portion of human genome. These sequences show high degree of polymorphism and form the basis of DNA fingerprinting. Since DNA from every tissue such as blood, hair follicles, skin, bone, saliva, sperm, etc. from an individual show the same degree of polymorphism, they become very useful identification tool in forensic applications. Further, as the polymorphisms are inheritable from parents to children, DNA fingerprinting is the basis of paternity testing in case of disputes. As polymorphism in DNA sequence is the basis of genetic mapping of human genome as well as of DNA fingerprinting, it is essential that we understand what DNA polymorphism means in simple terms. Polymorphism variation at genetic level arises due to mutations. Recall different kinds of mutations and their effects that you have already studied in Chapter 4 and in the preceding sections in this chapter. New mutations may arise in an individual either in somatic cells or in the germ cells, cells that generate gametes in sexually reproducing organisms. If a germ cell mutation does not seriously impair individuals' ability to have offspring who can transmit the mutation, it can spread to the other members of population through sexual reproduction. Allelic, again recall the definition of alleles from Chapter 4, sequence variation, has traditionally been described as a DNA polymorphism. If more than one variant allele at a locus occurs in human population with a frequency greater than 0.01. In simple terms, if an inheritable mutation is observed in a population at high frequency, it is referred to as DNA polymorphism. The probability of such variation to be observed in non-coding DNA sequence would be higher, as mutations in these sequences may not have any immediate impact in an individual's reproductive ability. These mutations keep on accumulating generation after generation and form one of the bases of variability polymorphism. There is a variety of different types of polymorphisms ranging from single nucleotide change to very large-scale changes. For evolution and speciation, such polymorphisms play a very important role, and you will study these in details at higher classes. The technique of DNA fingerprinting was initially developed by Alec Jeffreys. He used a satellite, DNA, as probe that shows very high degree of polymorphism. It was called as variable number of tandem repeats, fifth and TR. The technique, as used earlier, involves southern blot hybridization using radio-labeled V5 TR as a probe. It included 1. Isolation of DNA, 2. Digestion of DNA by restriction endonucleases, 3. Separation of DNA fragments by electrophoresis, 4. Transferring blotting of separated DNA fragments to synthetic membranes such as nitrocellulose or nylon, 5. Hybridization using labeled VNTR probe, and 6. Detection of hybridized DNA fragments by autoradiography. A schematic representation of DNA fingerprinting is shown in figure. The VNTR belongs to a class of satellite DNA, referred to as mini-satellite. A small DNA sequence is arranged tandemly in many copy numbers. The copy number varies from chromosome to chromosome in an individual. The numbers of repeats show very high degree of polymorphism. As a result, the size of VNTR varies in size from 0.1 to 20 kb. Consequently, after hybridization with fifth thin TR probe, the autoradiogram gives many bands of differing sizes. These bands give a characteristic pattern for an individual DNA. It differs from individual to individual in a population, except in the case of monozygotic identical twins. The sensitivity of the technique has been increased by use of polymerase chain reaction, PCR. You will study about it in Chapter 9. Consequently, DNA from a single cell is enough to perform DNA fingerprinting analysis. In addition to application in forensic science, it has much wider applications such as schematic representation of DNA, fingerprinting. Few representative chromosomes have been shown to contain different copy number of VNTR for the sake of understanding different color schemes have been used to trace the origin of each band in the gel. The two alleles paternal and maternal of a chromosome also contain different copy numbers of VNTR. It is clear that the banding pattern of DNA from crime scene matches with individual B and not with A. In determining population and genetic diversities, currently, many different probes are used to generate DNA fingerprints. Summary: nucleic acids are long polymers of nucleotides. While DNA stores genetic information, RNA mostly helps in transfer and expression of information. Though DNA and RNA both function as genetic material, but DNA, being chemically and structurally more stable, is a better genetic material. However, RNA is the first to evolve, and DNA was derived from RNA. The hallmark of the double-stranded helical structure of DNA is the hydrogen bonding between the bases from opposite strands. The rule is that adenine pairs with thymine through 2 H bonds and guanine with cytosine through 3 H bonds. This makes one strand complementary to the other. The DNA replicates semi-conservatively. The process is guided by the complementary H bonding. A segment of DNA that codes for RNA may, in a simplistic term, can be referred as gene. During transcription also, one of the strands of DNA acts a template to direct the synthesis of complementary RNA. In bacteria, the transcribed mRNA is functional, hence can directly be translated. In eukaryotes, the gene is split. The coding sequences, exons, are interrupted by non-coding sequences, introns. Introns are removed and exons are joined to produce functional RNA by splicing. The messenger RNA contains the base sequences that are read in a combination of 3 to make triplet genetic code to code for an amino acid. The genetic code is read again on the principle of complementarity by tRNA that acts as an adapter molecule. There are specific tRNAs for every amino acid. The tRNA binds to specific amino acid at one end and pairs through H bonding with codes on mRNA through its anticodons. The site of translation protein synthesis is ribosomes, which bind to mRNA and provide platform for joining of amino acids. One of the rRNA acts as a catalyst for peptide bond formation, which is an example of RNA enzyme ribozyme. Translation is a process that has evolved around RNA, indicating that life began around RNA. Since transcription and translation are energetically very expensive processes, these have to be tightly regulated. Regulation of transcription is the primary step for regulation of gene expression. In bacteria, more than one gene is arranged together and regulated in units called as operons. Lac operon is the prototype operon in bacteria, which codes for genes responsible for metabolism of lactose. The operon is regulated by the amount of lactose in the medium where the bacteria are grown. Therefore, This regulation can also be viewed as regulation of enzyme synthesis by its substrate. Human Genome Project was a mega project that aimed to sequence every base in human genome. This project has yielded much new information. Many new areas and avenues have opened up as a consequence of the project. DNA, fingerprinting, is a technique to find out variations in individuals of a population at DNA level. It works on the principle of polymorphism in DNA sequences. It has immense applications in the field of forensic science, genetic biodiversity, and evolutionary biology.